day, everybody. My name is Ed Mills. I work at Cytel, and we're delighted that you're able to join us today for a webinar co-hosted by Certara with Cytel on this extremely important topic of COVID trials. We're going to talk about some of the COVID trials that are currently going on. We're going to talk about some of the issues that will assist you in planning perhaps your own trials or finding out about trials going on that are relevant to you. And we're going to talk about why COVID trials have pushed the agenda in terms of methodology related to clinical trials. I'm joined by Srin Murthy, who is also in Vancouver. He is an associate professor at University of British Columbia, a critical care physician. He is co-chair from the Canadian side for the Solidarity trial from the WHO. He is also a co-investigator with the Remap CAP trial. I think you're going to be very interested to hear from Srini about those ongoing trials. I'm also joined by my colleague and good friend, Craig Rayner, President of Integrated Drug Development at Certara, who'll be talking about pharmacology related to choosing interventions. By my colleague, Louis Dron, who is a director at Cytel. He'll be introducing you to the clinical trial tracker, and my colleague, Christian Thorland, who is also at Cytel and a professor at McMaster University. So Srin, I'll hand it over to you.
Thanks, Ed. And so I'll talk about two trials that are ongoing and actively recruiting in a variety of places. And so there's two sort of sequential slide decks. The first we'll talk about Remap CAP, and the next we'll talk about Solidarity. I'll speak for about 15 minutes total. I think we'll have time for questions. And so Remap CAP is a randomized, embedded, multifactorial, adaptive platform trial for community-acquired pneumonia, which is what it was originally described, but obviously we've pivoted specifically for COVID-19. Next slide, please. As obviously many people know, traditional RCTs are serial testing of single disease. Next, with an antibiotic trial testing 3 agents and then subsequently getting an outcome. Next, or a macrolide trial testing 2 agents and subsequently getting an outcome. Next, or a steroid trial getting 2 outcomes and subsequently getting an outcome, all in sequence. Next, a Remap, which is a randomized embedded multifactorial adaptive platform trial, is a self-learning system whereby we can test all 3 of these things simultaneously, assess outcomes, and through adaptive randomization, continue to provide the best possible care within the framework of a clinical trial so that we're constantly learning. And with that, we're constantly adapting to emerging evidence. And so currently we're recruiting over, and this is out of date, this is a couple of months ago, I think we're up to, 100 sites in 14 countries right now, primarily in Canada, the EU, UK, Australia, and now increasingly in the United States. Also in Saudi Arabia and Singapore and Brazil, rolling into Remap CAP as well. Our website is remapcap.org, which has an updated list of enrollments and countries that are activated. And so the randomized component, obvious to this audience, embedded. So it's a very straightforward clinical trial design whereby clinical staff themselves can randomize, or research coordinators if they're available, and then deliver intervention using the usual clinical processes. We'll talk about how some centers and infrastructure has been adapted to embed things within the electronic health record directly. Multifactorial, we discussed. The adaptive component is that there are frequent Bayesian adaptive analyses throughout the course of the clinical trial with response adaptive randomization embedded within each of those intervention arms, with domains and interventions being added in as new interventions become available or biologically plausible. And it's a platform trial, so this is for severely ill patients currently, so all patients with CAP can be conceivably included. And so for this pandemic, we have designed a specific pandemic strata, which was our original intention when we designed the trial a number of years ago. And this was formally activated on March 13th, 2020. And so what that pandemic strata does, it changes the eligibility criteria, changes the primary endpoint from a distal mortality endpoint to something we'll speak of, more proximal, changes the statistical model, changes the frequency of the response adaptive randomization, and integrates new pandemic specific domains specific to COVID-19 directly. And so currently our eligibility is, does the patient have clinically suspected or proven pandemic infection? And we have various frameworks as to what that means. Namely, testing is being done is not sufficient. You must think it is likely. And so patients who have testing done purely for ruling out would not qualify. This is a likely type diagnosis using clinical judgment along those lines. And the way the analytic models are set up is that we will end up stratifying by patients who have actual confirmed disease, and we'll analyze patients who have received a couple of doses of, say, the targeted intervention, and patients who do not end up having the disease despite having clinically suspected disease. We changed our definitions of what an ICU is. Previously, we had an organ function specific support criteria as well as an ICU support, given the changing and dynamic landscape of where patients are being taken care of during a pandemic. We described it mostly as what they are receiving as defining what an ICU is. And so wards conceivably, which provide ICU level interventions, could be conceivably an ICU. We changed our primary endpoint from a distal mortality endpoint to a days alive and out of ICU at day 21, acknowledging that the definition of ICU is not a geographic one, but is an organ function specific one. And we censor this at hospital discharge. We will continue to get follow-up until day 90 as part of our secondary outcome from our original design, but this is not part of the primary endpoint that's driving the primary analysis and the response adaptive randomization. Our statistical model, it's complex, and it's run through Barry and Associates in Austin, Texas. I'm not going to go into all of the analytic strategies over the course of the next couple of minutes, but briefly, domains that are considered relevant to the pandemic are analyzed using a separate statistical model compared to domains that we do not think are considered relevant to the pandemic. And so antibiotics, for example, bacterials, are not considered within that pandemic specific model, but antivirals specific to COVID-19 would be. With the response adaptive randomization occurring separately for patients with and without proven pandemic infection. So we can continue to enroll non-pandemic patients within this learning system during the pandemic. And in those slides, you see how that model works. And so PIN-SNP is pandemic infection, neither suspected nor proven, and PISOF is pandemic infection suspected or proven, and how they would integrate with each other across different models. One model within a pandemic, one model between pandemics. And we'll apply the knowledge from the pandemic model directly during this pandemic. So like I said, the model evaluates all pandemic domains, evaluates patients who are confirmed COVID-19. We've changed the stats trigger from 0.99 to 0.95 for our probability of superiority, specific to this pandemic and specific to the idea that we want to get knowledge out faster, acknowledging that obviously there's some criticism of that decision making. The RAR is driven off the confirmed COVID-19, like spoken about earlier, and assesses safety in suspected patients who turn out to be non-COVID patients, as described earlier. Currently, we're evaluating corticosteroids and macrolides from the original community-acquired pneumonia domains in the pandemic model. We're also adding in specific COVID-19 domains and antiviral domains, starting with Kaletra and hydroxychloroquine, but options for adding in an immunomodulation domain with those listed agents available currently, with different interventions in different regions. So that in the UK, for example, they're evaluating those IL-6 inhibitors. In the EU, they're evaluating anakinra. And so different regions can evaluate things in their own way, and that can contribute to their model learning. Anticoagulation is being evaluated, vitamin C is being evaluated, and convalescent plasma currently is being evaluated across the platform for a rapid learning of whether these interventions are useful or not. The consent processes during a pandemic are complex, but we are doing sort of creative things with phone and electronic confirmations and verbal consents as appropriate, depending on the jurisdiction and the region. There's a DSMB, which has been granted more flexibility and has the ability to ask for additional analyses and has the permission to contact public health authorities directly with any information that they believe is important to inform the public health response. And we've organized these relationships with officials at the WHO and other agencies. I think Dr. Derek Angus is speaking in a couple of weeks about how he's embedded this in the United States across his electronic health record, across the UPMC hospitals, effectively a learning while doing clinical trial of COVID-19. It's not a vaccine. That is a misnomer. I guess that's a media thing. But he has done something very creative where he's integrated randomization into his Cerner build to make it part of care. I also want to speak briefly about Solidarity in the last 3 minutes. Solidarity, many of you have heard about, is a World Health Organization platform trial. Dr. Tedros announced it on March 18th with the idea that multiple small trials with different methodologies may not give us the strong evidence we need about which treatments help to save lives. And I think that's self-evident based on some of the descriptions that are going to happen after my presentation about the number of trials that are ongoing. And so with that, the WHO used its convening and collaborative power to engage the global community to do one trial and learn as quickly as possible. So currently it's labeled a mega trial with the aim of enrolling tens of thousands of patients with a common analytic set and a common randomization schema. Currently, its PICO is that populations of hospitalized adults with confirmed COVID-19 randomized to one of those 5 therapies with a common standard of care, optimized supportive care arm with the primary outcome of hospital mortality. So this is a unique trial in that there are obviously no sample size and no pre-specified stopping rules. The DSMB, which is a global DSMB, has been given a large amount of authority and power to decide as to whether or not a targeted intervention meets criteria for stopping, and then that will be disseminated widely. They'll have frequent interim efficacy analyses, but no necessary set rate of those interim analyses. It's a very straightforward design at the site level, so it's embedded within care as much as possible. With this very rudimentary baseline demographics, minimal baseline severity, the drug given, and outcome at discharge. It is an adaptive trial with frequent interim analyses, and then we'll adapt to new interventions as they become available. So that if a new drug or a new intervention is deemed reasonable, it will be either added in as a separate randomization arm or as a separate randomization altogether to have multiple levels of randomization. And other agents will be either dropped for harm or futility, or if they are proven effective, integrated into the standard of care arm in real time within the scope of the trial. Currently, it's active or becoming active in 93 countries around the world. And it's sponsored by a number of different countries and academic institutions. And so it's a unique trial that's in countries across the world. And it's going through the regulatory approvals everywhere currently. As of, I think, yesterday, there have been about 17 or 1,800 patients randomized thus far into the schema, and data should be accruing and reporting as conclusions are reached.
Thank you very much, Sven. That's fantastic. I will let the audience be aware there's in the GoToWebinar control panel, there's a section where you can enter a question in, and hopefully if we have time at the end of all of the speakers, we'll be able to get to answering some of those. And if we don't have time, we'll reach out to you afterwards with some answers if we've got those answers. So the next speaker that we've got is Dr. Craig Rayner, President of Integrated Drug Delivery at Certara, also a professor at Monash University in Melbourne, and he's coming to us from Melbourne, Australia tonight. Craig?
Great, thanks, Ed. In our response and preparedness planning for medical countermeasures development, that being vaccines and drug therapeutics, for infectious diseases outbreaks, we tend to cycle between panic and also apathy in the inter-pandemic or inter-epidemic times. So when we consider where we kind of been, we've had Ebola, Zika, H5N1, pandemic H1N1. Our experiences with COVID, sadly, really represent Groundhog Day. So in our response, is that clicking? It is, okay, yeah. So earlier this month in a joint webinar with the Gates Foundation, I described the 5 lessons that we learned from our past experiences in trying to develop therapeutics within pandemic circumstances. And they were, know what you're up against. It's really, really important to understand how challenging it is to develop therapeutics within a pandemic. And I think you've seen some of the creativity that Sren and many people within the medical and community are actually trying to apply to help us be able to move things forward. You also have to begin with multiple audiences in mind. So we've got people who are procurers of medicine, frontline physicians, we have patients, we have people who are investing, public health officials. There's lots of different stakeholders that need to be considered. In the emergency response situation where we are right now, you actually have to have a roadmap. You also have to have highly integrated and agile teams and global teams. And importantly, with the right mindset and the right culture in order to be able to develop medicines and progress medicines and therapeutics in the circumstances within a pandemic. You cannot do checkbox drug development. You have to be very smart. You have to be very creative. We've already heard about that. And you can't do this alone. And it really requires a coalition, and not just a coalition of the willing, but a coalition of the competent. I'd like to briefly recap on just a few of these. And again, these are the ones that I chatted about at a prior webinar, but those being, really knowing what we're up against. I'll touch on, being smarter in the way in which we approach drug development, and then a little bit of a call to action and collaboration. So we can move to the next one now. So the best analogy I have to describe responding to the COVID situation is really like weather forecasting. Firstly, the public health officials, the therapeutics and vaccine developers, our clinicians on the front line treating patients, and those engaged in public policy are all doing their level best at forecasting all things related to COVID in times of great uncertainty. And the second point that I would say is that like the weather, everyone has an opinion on it. And given the large amount of uncertainty, pretty much anyone can position themselves as an expert commentator in any particular area and impose decisions that may not necessarily be aligned with the relevant experts' advice. So look, as a drug developer, developing therapeutics in a preparedness or a peacetime mode is hard enough, but doing it during a pandemic is very, very tricky. Investigational and repurposed candidates actually bring their own unique challenges, and each compound has to be evaluated individually. Of course, an investigational therapeutic may seem further away from the clinical application at first, but just because a drug is approved doesn't actually mean it's fit for purpose. I said this at the prior webinar, is it really logical to think that a dose regimen that was developed for HIV or malaria or rheumatoid arthritis or other parasitic infections will be fit for purpose for an acute virus infection in the lungs where achieved concentrations at the site of infection are 10 or 100-fold lower than results that we're actually getting from a petri dish? And just because a drug is available, it doesn't mean that we can ignore the translational pharmacology that sits between the petri dish and administering a potentially safe and effective dose for patients. Because if we do, we actually push all of that risk forward to the patients that we're actually wanting to help. So of course, it doesn't mean that we have the luxury of developing medicines on the traditional timescale of years. In fact, we all have to go much faster, but we just need to know that there are some shortcuts that should and others that should not be actually taken to get the right drug at a safe and effective dose to patients as quickly as possible. So knowing what we're up against, and I guess we're keeping with this weather analogy, as a drug developer, I feel that we're in a yacht race in a hurricane. If we can move to the next slide. So in the chaos of COVID, I wanted to raise a critical question. Who's actually thinking about optimal dose regimen? So why optimal? Well, look, every milligram above an optimal dose is less therapeutic for others. It's increased manufacturing capacity. It's additional pressure on the supply chain. It's challenges for actually getting medicines actually deployed. And it's also the increased risk for safety and tolerability issues. And at the same time, every milligram below optimal is giving the virus a chance to outsmart our interventions. So again, who's actually thinking about optimal dose? So of the more than 535 clinical trials, when I looked at it, and I'm sure it's more, we'll hear from other colleagues from Cytel today, it's really difficult to find many that are looking at dose regimen finding in design. So currently the optimal dose regimens for COVID will need to be deconvoluted from existing trials, giving many of the studies underpowered. They include duplicative regimens, non-pullable endpoints. I have little faith that pulling from the existing trial database will be particularly convincing. I think we heard, the WHO Solidarity Trial, Remap CAP. These are wonderful advances, and I think through their efforts will certainly offer some increased ability to look at this, but will ultimately be limited to the study doses that are evaluated within those frameworks and the appropriate meta-analytic methods that are applied to be able to understand what is the appropriate regimen, but as I'm trying to touch on, is the appropriate or optimal dose. So anytime we're actually talking about the science of dosing, we're talking about clinical, translational, and quantitative pharmacology. And in industry-driven drug development programs, these disciplines are critical to inform the doses for evaluation in RCTs. This is the enormous amount of effort that sits between the Petri dish and the determination of the dose to be evaluated in an RCT. So right now, I feel that the weakest link in the global drug development response to COVID is this apparent skipping by some of these essential pieces to get the science right around the dose regimen. It's a false economy. I think we're seeing it already with the volume of trials activity, duplicity and wasted efforts. We're seeing it with repurposed drugs naively being administered at doses that they should not be dosed in patients and are actually putting patients at risk. And ultimately, if we learn a repurposed drug is effective for, say, in prophylaxis against COVID, without having strong insights into what the optimal dose regimen will be, how do we respond? Is it just simply more trials to work out the next dose? How much drug should be procured, deployed to whom, and who misses out? So let me double click a little more on the importance of not skipping the essential pieces and to get that science right around the dosing regimen as we're going through. So we think of drug development process for COVID as a cycling race. We have a COVID-19 or a C19 peloton who moves to various checkpoints and ultimately to the finish line of delivering a safe, effective, optimal dose regimen of a therapeutic for COVID. The first leg of the race is really demonstration of pharmacological plausibility. So that's the mechanism of action, preclinical activity against the target, which could be SARS-CoV-2 or the host target as relevant, and establishing proof of hope. So proof of hope is the expectation that pharmacologically relevant concentrations at the target can be achieved in humans, that a therapeutic index exists, and no showstoppers exist for other clinical pharmacology considerations. So assuming that we successfully traverse that first checkpoint, the peloton then moves to the second leg of the race, which is really the clinical proof of concept and optimal dose determination. This is the demonstration that the pharmacology translates to the clinic and a dose rationale that can be established. And assuming success, the riders move on to the last stage, clinical confirmation and the real world application, which is again, the RCTs, the evaluation of real world evidence, and of course. Let's take, for instance, the well-publicized example of ivermectin, where Kelly and colleagues reported the in vitro activity of ivermectin against SARS-CoV-2 cells in serocells. The study described the pharmacological plausibility for ivermectin having activity against SARS-CoV-2. However, didn't talk about the translation of the concentrations in that in vitro system tested to the clinic. Based simply on published PK information at typical single doses of 150 microgram per kilo, plasma Cmax values would at least be 40 to 60-fold below the in vitro EC50 was tested within the within these in vitro assessments. So let's take, for instance, then the application of translational pharmacology. So using PBPK modeling, physiological-based pharmacokinetic modeling, we also showed that even at very high dose of ivermectin regimen of 600 microgram per kilogram per day for 3 days, the in vitro EC50 is still more than 20-fold higher than the highest anticipated lung tissue concentrations. Now, unless new evidence emerges that suggests clinically relevant concentrations with an appropriate safety window can be achieved, which is not the case currently, clinical investigations should not be advocated or supported. See, our time and resources are very precious now. We must apply our resources to those compounds that are most likely to succeed against COVID. And the burden of proof for moving drugs to the clinic has to include consideration of these translational pharmacology principles, because if we don't, we just move the risk forward to patients. Now, science is littered with in vitro promise and clinical failure. That's not new. And this should be a good example where a key method in the translational pharmacology toolbox of physiological-based PK models can be applied, and the peloton should be stopped at that proof of hope checkpoint. So look, we of course cannot be sequential in the way in which we inject translational pharmacology principles into the COVID response either. We can't be applying this. These are not standard times to apply standard industry approaches. So what I'm saying is that improved same in the way we do drug development is simply not good enough. Federated approaches, as we've heard, like the master protocols, Remap, solidarity, these are really very important ways to drive harmonisation of efforts and efficiencies, particularly around aligning information collected and on endpoints. And these are very, very important ways of doing things. But we should also augment these approaches at every opportunity and use our existing translational pharmacology toolkit to create an ecosystem with other types of information and be making decisions on totality of evidence. So just a few examples where these translational pharmacology approaches may add value within the current COVID response include. So simply adding pharmacokinetics to the existing studies. These support PK/PD modeling exercises, which can be done in parallel. They can help us generate dosing guidances for other populations, the children, elderly, people of differing body size, bringing into account ethnopharmacology issues, conmeds, the setting of renal or hepatic impairment or the critically ill, or altering dose requirements at differing stages of the disease and so on. Performing specific small-scale clinical trials where PK/PD is intensively explored can be quite powerful in teasing out drug effect and support dose determining optimal dose. This can be expanded into the highly efficient clinical trials principles that have been discussed today in umbrella designs as an example, where you can have combined PK/PD umbrella studies with real-time readouts and the application of modeling and simulation that's actually working in parallel. And we can certainly improve even the quality of the emergency use or compassionate use programs that are going on, just to gather PK/PD information that may help earlier decision-making on drug effect and dose. And there are actually some good examples in the literature which have demonstrated this. Pharmacoepi models that insert drug pharmacology variability more explicitly into the epidemiological approaches to guide impact not only on the individual viral shedding characteristics, but also more so in terms of the impact of spread. It's very important to be thinking about pharmacological variability when we're thinking about these aspects. Real-world registry studies, they can be used to tease out associations around drug regimen effectiveness via targeted deployment of registry regions and sites to enrich data sets with specific regimens, and of course the plethora of modeling and analytical methods that can be used to cater with the grey data at a single trial or across a data ecosystem. And yeah, so I guess what I would say in closing, and I'm on to the next to last slide, it's important to stress that the application of these translational pharmacology principles is not to cut corners. It's not to avoid critical or objective thinking. It's really to support, augment processes. It's to increase parallel processing and make the most of the available information content to cut time and accelerate the development and access of critical medicines to treat COVID. And look, the final thing I guess I'd really like to bring up is, for me, the final lesson. In order to adopt these translational pharmacology principles and increase scientific rigor to conquer COVID, with the funding from the Gates Foundation, we established the COVID Pharmacology Portal. And this will link to other important resources, including the trial tracker from Cytel. It's a pre-competitive website that seeks to provide a platform to share knowledge, collaborate and invite others to contribute. There we actually provide in silico tools to support clinical trials design, forums to discuss issues relevant for advancing therapeutics for COVID. And again, just underscores the final lesson that I wanted to share. The only way we can actually conquer COVID is together. So the future depends on what we do in the present, and let's just hope that our COVID legacy will be improved global preparedness for the next disease X. So with that, I'd like to conclude and hand over to the rest of the team.
Thank you very much, Craig. And let me express my gratitude to you for being involved in many of the trials for COVID. We need people like you who've given a lot of deep thought to this, and also a lot of experience with diseases that resemble this. So our next speaker is Louis Dron from Cytel, who will introduce to you the clinical trials tracker. Louis?
Yes, thank you very much, Ed, and thank you very much to the two previous speakers. It's been fascinating to hear about so many of the aspects of the work which has been going on in this disease. And so what I'd like to talk about today is some of the activities that we've been undertaking in terms of trying to understand what's happening globally in terms of interventional trials for COVID-19. And so to do this, we've generated an online website which we'd very much encourage everyone to go and have a little play around with after this call, and feel free to send feedback on it as you do so. And I'd also like to, at the very beginning, just to express our thanks to many of the different groups and partners which have helped out in the development of the website, for the Bill and Melinda Gates Foundation, FAST Grants, all of the wonderful team that we've been working with internally here, but also anyone externally that's given us some advice, some feedback, that's highlighted interesting things that we weren't aware of. We've actually had feedback from almost all continents across the planet at this point, which is fantastic. So just to give a brief sense at the beginning here of what the website represents, we're trying to track interventional clinical trial activities for patients with COVID-19. And so to do this, we're taking a number of different data sources which are representative of global trial efforts here. So we have 8 registries which we look at. Each of these registries will be recording different amounts of information about registered and planned clinical trials. For example, how many patients they're planning to recruit, what treatments they're interested in, what dose they're interested in. We synthesize all of these together and we pull them into one centralized repository. We use a mixture of semi-automated methods and traditional manual review to add a little bit of extra context. So for example, we make sure that if you have a trade name of a treatment, that's converted to a sort of standardized name here, so that people, when they're searching, can make sense of the registries and what it is they represent. If we could go to the next slide, please. So we won't be doing a live demoing of this today. And again, the real principle here of the website is to be able to give to the research community the tools they need to do their research. But just to provide a very brief overview here, there's a number of different ways you can filter the trials and the activities which are going on around the world. So on the right-hand side here, you can see you can select by a number of different treatments, and this will give you just the trials, the unique trials, and where they're being conducted globally here. You could also filter by countries on the left-hand side. You can put in filters for whether you want actively recruiting trials, ones which are just planned. There are a very small handful which have completed, which are also incorporated on here. And what you'll get at the bottom is this table of results indicating a few of the key characteristics. They have links to the original registries if you want to dig into the details a little bit further or get a bit of a deeper understanding of what's going on. And you're also able to download all of the data and make whatever sense of it that you want to. And so what I want to cover a little bit today, just towards the tail end of this discussion, is some of the trends which we're seeing so far. Next slide, please. So I think this really speaks to what we've covered in the previous discussions here, but this puts a few numbers behind it here. So of all of the 590 trials which we have identified so far, planned or actively recruiting, around half of them are incorporating a planned sample size of 100 or less. And the lion's share of these are also specifically two-arm clinical trials. Now, it's not that you can't necessarily get meaningful research questions answered with such a sample size, but it's indicative of this shift for a potentially fragmented landscape with a number of different groups. There are a number of different reasons why people may not be able to be in contact and have duplication of efforts, but there is certainly a trend here towards these smaller trials being conducted. And there's also certain treatments which may or may not be surprising to those who follow the space, which are massively represented in a potentially disproportionate way. So we at the moment are tracking, trials incorporating either hydroxychloroquine or chloroquine. We've simplified them together for the purposes of data visualization. There's 118 trials utilizing one of these compounds, either as a single arm or as a component in a number of different treatment arms that they have here. And so on the right-hand side, you can see also the pace of change of this. And this has really jumped up in recent weeks. But then there's also been some rather interesting readouts from some preliminary evidence. And it'll be interesting to sort of see how this curve develops over time. Just by way of illustration, if every single patient is recruited in every single one of these trials and they reach their maximal sample size, it'll be approximately 195,000 participants who would have been treated with this one or two of these treatments. Now, obviously, some of these are going to be in different contexts, and I think Craig and Trin have both mentioned earlier, there's a lot of different, maybe there's different dosing regimens which are of interest. Maybe there's some different patient populations, but I still feel that perhaps with 195,000 patients, somewhere in there should be sufficient clinical activity to understand whether these treatments may or may not be effective. Next slide, please. So there's a lot of geographic variation, and I think there's perhaps parallels what you see in non-COVID-19 related trial activities, but it's interesting nevertheless for some unique characteristics to this outbreak. So the United States is actually the country with the second largest number of registered clinical trials. 47 are actively recruiting at the moment or have a trial site in that country. And you can sort of see on the right-hand side here the trajectory and the pace of change for which these are being registered. So that almost half of the current number are actually registered within the first couple of weeks of April to date. And again, here we have interesting breakdowns to do with treatments. This isn't to particularly harp on hydroxychloroquine, but it's such a standout feature that it seems like it should warrant some attention. A third of the trials registered in the United States incorporate at least in one component hydroxychloroquine in some form or another. So you can see here this sort of trajectory is massively increasing, and perhaps it will continue to do so, perhaps it will flatten out. One interesting counterpoint to this, next slide please, is in China. So in China, there are a very large number of registered clinical trials, 318 by our count at the last readout that we had here. And one interesting component of the registrations of trials in China is the sort of flattening out of the curve, resembling the reported number of cases there in China. So whereas in the US, more than half of those trials were registered in the first 2 weeks of April, there's only 9 trials added over the same time period in China, representing around about 3% of all of their registered clinical trials. So interesting parallels there, perhaps the reported number of cases. And the number of treatments is very variable here. That there are treatments accounted for in China which are not in many other countries for traditional Chinese medicine, for example. There's only 6% of those trials registered there are incorporating hydroxychloroquine. Again, whether hydroxychloroquine in that context was as part of a standard of care not recorded, hard to say, but it definitely seems to be receiving a little bit less attention. Next slide, please. So then thinking more broadly geographically across the world here, there's also a tremendous disparity in where trials are being conducted and registered at the moment. And I think Trin maybe alluded to earlier that this is going to be changing from the looks of the Solidarity trial sites. But as of today's date, this is the approximate distribution for a number of continents and subcontinents here. I think one component that's been sort of, relatively surprising and not surprising in some senses is the number of trial activities in South Asia and the African continent. So combined between these two geographies, there's less than 20 registered clinical trials. And in many countries in these regions have unique patient characteristics which may preclude those participants from evidence in trials being conducted in Western settings. So in a number of these contexts, there may be relatively high prevalence of tuberculosis, HIV. There may be high prevalence of malnutrition. And how all of these factors influence in COVID-19 remains to be unseen. On the other hand, it could just be that the representation of registered trials is a limitation of language barriers. We're only able to capture English and any other languages which we have local translational capabilities for. It may just be that researchers are finding it more important to get on with setting up trials rather than registering them on the clinical trial registry. There's a number of different reasons as to why that may be the case here, but it's an interesting visualization nevertheless. And so I think as we go forward, some of the key characteristics that will be fascinating to understand is how representative, as of today's date, are these registered trials when clinical trial data comes out? How many of these are going to be reached to completion? How many of these are going to consolidate into these larger clinical trials, as we've heard from earlier and we'll discuss shortly? It's all completely unknown, which is rather exciting in some senses to see how that's all going to come together. And we're going to be trying to integrate analytical capabilities to try and make sense of this, but that might be a topic for a seminar at another time. And with that, I'll hand over to Christian Thorne.
All right, thank you very much, Lou. So next slide, please. So I think I'll start with, so Craig was talking a bit about roadmaps for designing trials. And so just extending off of Lou's presentation of the COVID tracker, I figured it'd be a good idea to start out with how would we use this as a roadmap to design clinical trials. And Sharon's been talking about examples of platform trials and umbrella trials, how that kind of maps in to COVID. So if we try to look at the evidence of what it currently is, not geographically, but just overall across the world. And this is just the top 15 as in most frequently investigated treatments. Basically what you see here is any circle represents any of the top 15 treatments, and there's a few select combinations of them. Any line between two circles indicate that those two treatments have been compared in a clinical trial, or whether it be randomized or not randomized. We've only put in numbers here for those that have been compared to some sort of standard of care or no intervention, because quite frankly, it gets a bit too busy if we were to put numbers in for the head-to-head comparisons between the active interventions. But I think what this illustrates is that, and as you can see already from Lou's presentation, is that there is a lot of effort going on in interventional studies. I think we're currently over 600 registered trials. It's not all of them that are ongoing, but I think it's at least half of them that are currently recruiting. So if you're going into this thinking, well, wouldn't it be great if I did a randomized clinical trial? It almost looks like everything has been done already. But the fact is that when you dive deeper into it, and just like Lou was showing with some geographic regions that are maybe yet to be explored more thoroughly, when you dig deeper into which populations have received interventions, have been tested, and which have not, and what settings have that been done in, we may end up getting a different picture. Next slide, please. So I also just wanted to add to the richness of the evidence, how this may be overwhelming to begin with, but how once one digs a bit deeper into it, we start seeing a different picture. So here, what this graph is presenting is the expected total number of patients enrolled into any of the given top 10 treatment arms in trials that are set to complete this year, so in 2020. And as you can see, we're hydroxychloroquine, if all trials complete, and that may or may not happen, we're expecting to have about 50,000 patients having received hydroxychloroquine. And of course, that's a comparison to both standard of care and other health care interventions. So there's a little animation here, so NextBuild. Also, if we go and look at what clinical outcomes are being looked at, I think we can kind of see that all the cool outcomes are being looked at. So it's not like there's anything novel of clinical hard outcomes that aren't being investigated in the clinical trials. But if we start to split this up into subpopulations, we'll start to see a very different picture. Next slide, please. And so one of the ways that we, for example, have used this as a roadmap to guide what kind of trial we, or to guide the Gates Foundation in what kind of trials that they should be funding, is to try to split it up in different patient healthcare settings. So of course, we've done a lot of data extraction in the background, figure out what patient settings different trials are looking at. And what you can see from this slide is that the vast majority of trials that have been registered and are enrolling patients thus far are in some version of hospitalized patients, whether it's they're coming in the hospital, you're staying for a little bit, or you're more critical and you have to be put in intensive care. Whereas the evidence, the expected evidence over the next couple of months is perhaps a bit more sparse when we start looking at other very important populations like healthcare workers. How do you prevent them from getting sick? Outpatients that maybe are not presenting with severe symptoms and can just get a treatment and stay at home till they're healthy. But also just healthy, non-exposed or potentially exposed people. How can you use some of these treatments for prophylaxis? So one way that we used this was that, well, we figured that there are enough trials going on in hospitalized patients. Next slide, please. So the fact of the matter is that the treatment of COVID, it's a bit like about a month ago, there was a lot of talk about triaging these trials. So first you're exposed, perhaps you get a positive test, you're not so much at risk, you're healthy, young, no comorbidities, or maybe you're a higher risk patient and then you should be treated differently. So one of the trials that we've helped design, which is funded by the Gates Foundation, ended up following this type of triage design, where essentially you have multiple populations, you have sub-trials of exposed or likely exposed patients, so there's a prophylaxis component. You have patients that are confirmed COVID positive, but they're low risk, and you have patients that are confirmed COVID positive and they're considered high risk. And if you start with exposed patients, they can progress into being positive later. So if they are enrolled as exposed patients, perhaps later in the trial, they're actually COVID positive. So you kind of capture a much, much broader population. And at the same time, what this trial does is that it adds to the evidence that might be sparse. And the last component then is that if at one point your COVID progresses to severe symptoms and you end up being hospitalized, then through collaborating with other trials, then you can be simply transferred to a collaborative trial. Next, please. Next slide, please. The previous one, the one with the table. Okay, so just to put this in PICO's table format, because that's a nice way of looking at the study designs. This ended up being a dose response study for hydroxychloroquine, but in principle, it could be any intervention that's in there. So this is what that trial design would look like, and it's one example of one of the, at least, attempts we've made to be a bit innovative and adapt to the situation. Next slide, please. Next, thanks. Okay, so there's a couple of builds in here. So again, if we go back and look at the expected cumulative evidence in terms of the total number of patients being enrolled to any given treatment arm, there are some that have a lot of evidence, and there are some that maybe have less expected evidence over time. Next build. So if you are planning on, let's say you're planning on doing a hydroxychloroquine and azithromycin combination trial, and you started that a month ago, well, now NIH just started advising against that. And it'll be interesting to see what happens to hydroxychloroquine, azithromycin individually and in combination. But at this point, I think as a clinical trialist, as an investigator, you may want to think about adding a different intervention to your trial if you already have a setup for your clinical trial. Next build. NextBuild. An alternative way to approach this is if, for example, you're planning on doing a stem cell therapy, the cumulative number of patients expected for stem cell trials is not as large. And what you might be doing in terms of planning your clinical trial is to simply say, well, we're expecting a certain amount of patients to be enrolled in other trials over time. So it would make sense, so NextBuild, makes sense to look at the external evidence over. Thanks. So it makes sense to look at the evidence over time. And so, for example, one thing one could do is to build in instead of just looking at interim data, which is very common in adaptive clinical trials, also looking at external data and perhaps analyzing with Bayesian predicted probabilities, or just looking at the intervention effects that have been proven in other trials thus far. What is the probability that our trial is going to succeed? Is it going to complement the ones that are already out there? Is there a need for reassessing the sample size? Perhaps we'd assume the 40% relative risk reduction to begin with, but the latest evidence has shown it's not that large. So there are all kinds of opportunities of taking all of this trial data and mapping it quite simple to really fit the needs of the individual trials that you're planning and running. Next slide, please. So I'll conclude with a bit of a comment on the collaboration that's been going on with COVID trials. Here's an overview of the number of non-COVID platform trials, basket trials, and umbrella trials, so essentially master protocols that have been registered and are now ongoing over the years. And so you can see that over the last almost 2 decades, there's been about 90 of them. NextBuild. The issue, though, is that a lot of the umbrella and basket trials, particularly, are quite small in sample size. So they're not really these grand master protocols that people are talking about that can solve every single question on a specific disease. They're quite small trials, and in fact, what you end up with is probably only a handful, if not 2 or 3, of large platform master protocols that are ongoing, have been completed, or are currently enrolling. Next slide, please. And so if you contrast that with COVID, here's just an example of 5 master protocols that are already ongoing. Srin talked about 2 of them. There are a lot more, and we've gone through, I don't know how many 100 trials and trying to get the data into the COVID trial mapper, and I can tell you there are a lot more platform trials and basket and umbrella trials out there. Next slide. And so I think just for the last thoughts to leave with you is that you think about how much has been accomplished in such a short time. It's essentially just been a few months where the clinical trial world has been able to establish more than 10 or 20 extremely innovative large master protocols, and there are a lot of smaller trials that are working together. Contrast that with not even 100 of these that have been done over the last 2 decades in other diseases. So my last comments or last thoughts would be, if you think about what's been done over the last couple of months, I hope that obviously we continue with this during this COVID pandemic, but also that we don't forget the momentum that we're currently on as this pandemic subsides and we go back to work on other diseases, because I think there's an immense amount of benefit to non-COVID patients that can be had in the future. Thank you.
Fantastic. Thank you, Christian. And yeah, I want to echo what Christian just concluded with. I've never seen the level of collaboration among scientists that we've observed in the last month. And let's hope that we can continue that level of collaboration. We've got 2 minutes left. I want to really quickly rush towards a few questions that we've got. So if I can ask my panelists to just give me 30-second answers, or max 30-second answers. We have a question, Srin, directed to you. Your studies in the pandemic, the choice of sites, are they driven by the incidence rates, or are they chosen for other reasons?
other reasons, funding availability, willingness to enroll, and recruitment rates also, namely incidence of disease. So all of those things are factors that go into where we recruit, but obviously we don't know where incidence will be in the next few weeks and months, and so we're hoping to scale out everywhere.
Thank you, Sven. Christian, quick question. Many trials are using either placebo or standard of care. Standard of care is going to change over time. How important do you think it is, this idea of borrowing a control group, if in fact the standard of care is changing?
I mean, I think that's quite important. That could be it could be a matter for a month or two that it could save to get an answer.
Louis, we've got a question asking about observational studies. Is there an effort to map observational studies the way you've done for clinical trials?
Sure. So we don't have any plans to have that incorporated into our data set at the moment, but there are a number of resources. And I sent a message to the person who sent the question, who are collating some of those other areas where people are looking more in detail at observational trials.
Okay. We'll take one last question, and we will answer everybody's questions by email. Perhaps to Srin, if the standard of care changes in a country, will this affect any of your trials?
So in theory, a standard of care with one of those agents should not change within one country without adequate evidence as part of a global trial. And so recommending hydroxychloroquine, for example, as standard of care should not happen.
Okay. So thank you very much, Sven. The slides will be available, or this webcam will be available in the next few days on here. Cytel and Certara also have other upcoming webinars directly relevant to COVID. I'd like to express my thanks to everybody on the panel today. Sven, Craig, Louis, Christian, thank you very much. And also thank you to the audience for attending today. We wish you all to stay safe.